Hi, everyone. Thank you for joining the team at Hands-On Labs today for our second webinar in our discipline-focused live demo series. Before I hand the mic over to our host, let me take a moment to review the webinar logistics and give you a brief introduction to today's event. We encourage our attendees to participate in the conversation by asking questions and sharing resources. Please use the chat function to join in the conversation. You'll find that at the bottom of your toolbar. If you have questions, add those in the Q&A box also on your toolbar. Dr. Duane will address these questions at the end of the demo so everyone has clarification on important topics and their questions are answered. All attendees will be entered into a raffle to win a $100 Amazon gift card today. We'll share the lucky winner in our follow-up email tomorrow, along with a recording of this webinar for you to reference moving forward. So, what can you expect to see today? During our Microbiology Lab Live Demo, you will get to delve into the world of microorganisms through our rigorous selection of labs hosted by our Chief Resident Scientist, Dr. Duane Cagle, and our in-house distance learning specialist, Pam and Roger. They'll be manning the chat and answering any questions that come through while Dr. D is presenting. Get ready to explore our rigorous microbiology curriculum from both an instructor and student perspective and experience the HOL Cloud, our advanced e-learning platform specifically designed to provide a tactile lab experience for distance learners. If you don't already know our host today, Dr. Duane Cagle is our Chief Resident Scientist and has been the brains behind Hands-On Labs curriculum development for the past 9 years. As mentioned, we also have 2 HOL distance learning specialists on the call today who will be manning the chat and providing helpful resources throughout the webinar. Without further ado, a warm welcome to Pam, Roger, and our main host, Dr. Duane Cagle.
Well, thank you so much for the introduction, Audra. And I'm so excited everyone took time out of their schedule today to join us to get a preview of our microbiology curriculum, materials, and our cloud-based lab manual hosting platform that we call HOL Cloud. So I want to kind of split the time equally between those 3 topics today. But more importantly than all that, I want to take time out for your questions. You know, I know being an educator and thinking about taking your microbiology course of all possible courses online is both intimidating and a little frightening. When we're just thinking about students culturing microbes, you know, outside the protected environment of our physical laboratories. So I know that you have some concerns, and I want to make sure we take plenty of time to address those as best as possible. So let's go. Good. We'll see how well my connectivity cooperates today. With any luck, this screen is going to show us the lab manual landing page for a student who is using our curriculum and our physical materials for their online laboratory portion of a microbiology course. So you'll notice on this student landing page that their lessons are divided between prerequisites and then the lessons themselves. And this is very, very important for micros. Certainly when we're talking about using materials safely, being prepared for working with cultures, and knowing what to do in case of an accident. So all of those topics are addressed in the prerequisites. I'm not going to take the time to actually go through those lessons as a group today. I'd rather go through the true curricular lessons that cover topics that we typically teach on campus. But I will just state that getting started, make sure a student knows how to use the interactive lab manual. Lab safety is a generic safety course with videos and phone numbers for the student. Using the vScope is unique to students using a virtual microscope, which we host compared to a real microscope, which is also available, and we'll talk a little more about both of those in a moment. And then most importantly, microbiology laboratory preparation introduces the students to the microbe cultures they'll be using, the proper incubation environment that they should seek out, and safety as specified for the procedures they'll be conducting throughout their course. Meaning, what happens if I spill a broth? What happens if my dog gets into an agar plate? So forth and so on. But without further ado, what I really want to focus on from the student standpoint of our demo today is an actual lesson. And I also want to show you some student resources above and beyond the lessons themselves. So students have a tab in the upper left-hand corner of their course homepage where they can access lessons, which we're doing now. They can take a preview of all the physical materials they're going to be using for the semester. Now, most of these materials we're providing in their kit, but if they want to just get an idea of where everything's used and which lesson it corresponds to before they even get started, they can review that. And also, we encourage them to do this, and we encourage them to review the student-supplied materials. So, you know, if they're running low on something that we expect them to provide, such as bleach, to kill off their cultures at the end of the term or at the end of a particular lesson, paper towels, things such as that, they can make sure they're prepared in advance. We do provide students in microbiology with supplemental documents that they can download and print and refer to at any time right there on their tabletop. And the most important of those documents is for students using a real microscope is to make sure they know how to assemble it and how to actually use that microscope well in advance. Also pouring agar plates. I'll confess, we've tried in the past to ship pre-poured plates, and that is not a good protocol. So we do require students to pour their own plates. However, they're poured on an as-needed basis with single-use pour tubes, as opposed to 1+ liter bottles of agar. So much safer to use just the agar needed for one plate. You don't have to worry about spills that way, or if you did spill, it's not enough material to harm you or your immediate environment. And then lastly, microbiology student FAQs. We know from doing this for 2 decades, common questions students have. So we provide them with an FAQ sheet in advance so they can refer to that easily without having to call us or reach out to us via email should they have a question. And then the virtual microscope. I'm not going to talk about it right now, but I'm happy to show it to you before the end of today's session. Again, all of our micro courses come with the option that you as an instructor decide on for your students to either use our virtual interactive microscope program or to have your students use a real microscope. All right, so let's go back to the lessons. I know most everyone at some point in their introductory micro lab courses has a session on staining. And so let's take a look at that and how we present the lab manual content to the students. You'll see that every lab is broken down into 3 sections: exploration, the exercises themselves, and then an evaluation, i.e. an exam. So let's dive right in. Now, more specifically, those 3 sections of the labs are titled at the header for each of their landing pages. So exploration, experimentation, and evaluation. Now, if you have a background in learning theory, you might recognize this structure as part of the 5 E's of science learning. It follows constructivist learning theory, and it simply just means that students learn by building on knowledge they already have. So if we break a lesson down into the exploration section, which is the foundation concepts the student must understand to be successful, and then we conclude the next section labeled experimentation, which, as you might imagine, is the actual laboratory hands-on procedures. And then we culminate all of that in an evaluation, where we give the student the opportunity to demonstrate what they've learned by completing the laboratory activities. Challenge number one, or at least number two, for your online laboratory students is time allocation. And you're going to see this gray time allocation bar at the beginning of every single lab section we look at today. You know, your students aren't showing up to campus for a predetermined amount of time at a certain day or days of the week. It is really on them to set aside sufficient time within their schedule to be successful with their laboratory activities. So we want to warn them of those time allocations throughout every lesson that they encounter in our lab manual. And then lastly, on this landing page for each section, the student is going to encounter the learning objectives that are unique to that section. Our learning objectives are modeled after Bloom's taxonomy, which again, conforms to constructivist learning theory. The learning objectives for the exploration section are rather lower level of comprehension, understanding. So they'll begin with verbs such as discuss, describe, or summarize, as we see here. Now, before a student starts covering this background or exploration content, they're going to take a pretest, and we call it a test your knowledge activity. And this is a formative assessment. It is designed solely to let the student know what they don't know coming into their laboratory. Meaning, have you covered this in your lecture already? And if you have, you know, did the student understand it? Or conversely, is the student working ahead and you haven't even talked about staining or anything like that in the lecture or morphology? And so if that's the case, then the student, you know, really needs to be aware of what they're about to encounter. Now, these are drag and drop questions always. This one we see it's a matching event, but it could be a sequencing, ordering, categorization question. But the takeaway is always drag and drop. Studies show that drag and drop are the most engaging type of interactive question. So we want to make sure the first activity that a student does is as engaging as it can be. Now, because this is a demo instance, I can't actually drag and drop these, but with any luck, we are going to enter a live student instance, and let's see if it'll let us do that. Oh, is it going to? Maybe not. All right, well, we tried. So goes technology. But if we had been able to drag and drop, we would then get immediate feedback once we make our answers, meaning the system shows us which selections we got correct and which ones we missed. And this allows the student to then be self-aware of any knowledge gaps that exist. Once a student takes the pretest, they do encounter the curriculum, the exploration background content. And we always design our exploration pages as manageable blocks of text accompanied by one or more images. Those images could be micrographs like we see here, or they could be actual graphics or photographs or diagrams. And we'll see examples of all of those in the upcoming pages. Very importantly, at the bottom of every content page is a question time box. And the question time box is designed to test the student's comprehension of the key concepts on that content page. So you might consider this, just-in-time testing for the student, meaning the student's not going to progress all the way through the background and then answer a few questions. They're going to be tested on a page-per-page basis. So they don't advance until they understand the content as it is presented. Now, again, if we were in a live student account here, as soon as I clicked on one of these selections, I would get feedback as the student, meaning the system would tell me if I got it right or wrong. Again, just makes me aware if I'm not paying attention as the learner that, hey, I need to go back and read this again. I'm not going to spend much time on the rest of these content pages other than to point out that they're all designed in the same fashion. Manageable blocks of text, always accompanied by images. And we'll see that, so forth and so on. Now, this background culminates with a description of gram staining and a schematic showing the student what happens in each of those steps. And yes, they will be performing these very steps in the upcoming procedures. And again, just like every page, there is a question at the end. The final page of this exploration section gives the student a report of all of their answers to all of the interactive questions. So they can just review once again before moving on to the procedures. And what we've learned, again, from 2 decades of doing this is students have to be prepared for their laboratory activities. And if they're not, those activities really don't mean a lot to the student because they can't connect the dots. You know, they can't see the big picture then. So what if they stained a sample on a slide? So what if they use gram staining? You know, why did one microbe stain pink and the other purple? You know, so it's just important that a student knows what's going to happen before they actually conduct these procedures and what it all means. So now the student's ready to begin the exercises themselves. Here again is that time allocation. It's very, very important for the students. In this particular lab, it's letting the student know they have to have active culture broths of the microbes that we've provided them. And if they don't, it usually takes them about 48 hours to develop those broths. So if they haven't done it in the past, they're going to be held up by 2 days now to get their results. Here are more learning objectives. You'll notice they're more advanced in their verbiage. They're more active. So they begin with verbs such as create, prepare, and compare. Here are the materials the students are actually going to be using. And I want to stop here at these cultures because I know you have questions as an audience today. I'm guessing, even though I don't get to see the chat that you guys are doing. But I'm guessing you've already asked, well, what microbes do students use? What do you supply them with? Well, we supply them with 3 pretty standard laboratory used microbes that are all non-pathogenic. So you can see them here. We provide students with E. coli, Saccharomyces cerevisiae, and Staphylococcus epidermidis. So, and again, all of those are guaranteed as non-pathogenic by the vendor that we work with. The 2 bacteria species are provided as lyophilized tablets. So they are inactive when they're shipped to the student, so it doesn't create any kind of shipping concerns with any kind of courier services or anything like that, totally safe. And then the students are provided detailed instructions for how to activate those cultures and maintain them throughout the teaching term. Microscopes, all of our curriculum, we already talked about a virtual scope versus a real scope, but our curriculum is microscope agnostic. Meaning, while we sell microscopes, if you choose for your students to use a real microscope, they are not forced to buy a microscope from our company. As long as they have access to a compound light microscope with an oil immersion lens, they are welcome to use that. So that's why we list microscope and oil immersion lens as a student supplied material. And then very importantly, here are the materials in their kit that they need to retrieve before they begin the procedures. And you'll see things like a negative stain, you know, the Congo red pipette, comes in a little sealed 1 mil dropper pipette. It's very, very safe. We don't have to worry, it's hermetically sealed. We don't have to worry about it leaking out in the kits or anything like that. And then obviously all the gram staining supplies at the bottom, which again are bagged separately from the other materials in the kit. So again, we don't have to worry about spills and whatnot. You'll also see things like blank slides, sterile swabs, so forth and so on. So once a student has all the materials, they are ready to begin the staining exercises. In this particular lab, the students are doing simple stains with the cheek cell slides, kind of like just general microscopy type usage there, just to get them used to doing this. And then in the final exercise, they are actually gram staining the provided microbes. But what I want to point out on this is that we write our step-by-step procedures very succinctly, never more than one or at most two sentences per step. And those steps always begin with a verb. So we really want to draw the students' attention to exactly what they are to be doing. And the most successful way to do that is to visually guide the students through each of those procedural steps. So you'll see after every 2 or 3 steps, there is a photograph using the materials that are in the student's kit in the way that they are prescribed to be used. So, you know, the student's drawing a target on the slide and they're pre-labeling the slides before they ever put a sample on them. This goes over heat fixing. We provide the students with a candle. It's a lot more safe than if they're using a burner flame or something like that. Also, a lot lower risk of them fracturing the slides from too much heat. You know, we're not assuming students have ever done this before, so we want to walk them through and have them perform it in the safest way possible. And then actually applying a direct positive stain here, and then showing them procedures for doing a negative stain, so the Congo red. At the bottom of every procedure page, there is both a blue data tab and a series of open-ended questions. The blue data tab includes all the student responses from the results of their experiment, meaning if they made slides, they upload photographs of their slides. And anytime a student makes a slide in Micro, we require them to upload a photograph of the entire slide before they look at it under the microscope. So that would be these 2 response panels. And if I was a student and this was an active account, I would just drag and drop my image file directly into this box and it would appear. And then also any observations or recordings I've made. So if I've been asked to pick the best magnification to see a cheek cell in this instance, I need to record that magnification in this data table, and I need to describe why I selected that magnification and what I'm seeing in the microscope field of view. And then, again, the actual magnified slide images, which we provide students with detailed instructions on how to take microscopic photographs using a cell phone camera. So there's no need for students to go out and buy any kind of special microscope camera. I also want to point out that this data tab both slides up and maximizes on the student's computing screen and then minimizes with just one tap. And this is very, very important for students using mobility devices such as tablets like iPads or Android devices. And indeed, our curricular platform, HOL Cloud, is compatible with small screen devices such as those. So not only can your students use a traditional desktop computer or laptop, but they can use a tablet if they would like for reviewing their lab content and even submitting their answers. Now, at the bottom of each of these exercise pages are open-ended questions. And these are designed to test the student's complete understanding of the concepts that have been covered in both the exploration section and then again in the laboratory procedures. It gives them an opportunity to relate their results to the overall concepts that are being addressed in this particular lab. And then, as promised, the second exercise, a little more detailed. This is where they are gram staining the cultures that we've provided them, the E. coli, the Staph epi, and the Saccharomyces culture. And yes, I know Saccharomyces is a eukaryotic cell and it's really not designed for gram staining, but we provide the student with a question about that too. So, we are aware, but we know students need as much practice gram staining as possible, and we want them to truly see the difference between that eukaryotic cell and how it really gives kind of weird results when it's gram stained versus the much more standardized results of the gram negative and gram positive bacteria species they're provided. All right, so that's an example of procedures in our microbiology experiment. Once again, they're very succinctly written for the steps, and they include numerous photographs to visually walk students through the procedures. We want to assure that students do not become frustrated with any questions they may have, because unlike their on-campus peers, they can't raise their hand and have their lab TA walk over and explain a procedure to them. So it is imperative that we visually guide students through the procedures. And if you ask me, that's students' #1 challenge. Not necessarily time allocation that we were talking about, but the fact that they can't ask a question and get an immediate answer like they could if they were on campus. All right, so let's look at the final section that a student encounters in one of our labs, and that is the evaluation section. Again, we have a time allocation. We have all the learning objectives from the prior 2 sections refreshed. And then we have the final evaluation. And the student is reminded when they start this that they're locked out of the content in their lab, and it is a timed event. Now, it's not a timed event from the fact that they only have so many minutes to complete it, but it's a timed event in that it records the amount of time they spend taking this, and that amount of time is visible to their instructor. So when we start the evaluation as a student, we encounter a dozen or so multiple choice interactive questions, but the interactivity is a little different this time, in that this time around, I don't see what I got right or wrong as soon as I click the button. I actually have to go through all the questions. And once I go through all the questions and submit my exam, then I'm provided with feedback of which ones I got right and which ones I got wrong. And not only does that feedback let me know where I'm at the end of the lab and how much I truly accomplished, but that feedback has details of actually why I missed something and where that information was originally presented in the previous 2 sections. So I can always go back and review. Not all is lost. I can't take the test again, but at least I'm aware as a learner of where that information was provided. And then lastly, extension questions. And extension questions, here's the one about the Saccharomyces cell. Extension questions are designed for students to apply the knowledge from their results and from the background section to address something that hasn't yet been presented to them. So in this case, the students asked to explain why their Saccharomyces cells might have been colored purple, pink, brown, or green. And yes, indeed, it's because it's a eukaryotic cell with the cell membrane wall structure very different than the bacteria that were provided to them in their kit. But anyway. And student has an authentication statement, and then lastly, they get a report of all of their submissions for this lab. As an instructor, you are also able to view the report, and you're able to grade it however you choose. And we will take a look at our grading tools here momentarily. But this concludes the student view of one of our labs. And it's always best practices to stop and have myself take a 2-minute breather right now. But also, I know my team has been answering your questions as you've entered them thus far. But if my team has seen any questions that they're not comfortable with answering and want to forward those verbally to me in the next 120 seconds, I'll do my best to get you some very quick answers before we move and take an instructor's view of one of our labs. And the tools we make available in our interactive lab manual to facilitate how an instructor teaches the laboratory portion of their online course. So if there are any outstanding questions, panelists, please let me know.
Dr. D, we have amazing engagement going on here, dozens of questions, but I've been collecting a few that Pam and Roger want you to sort of touch upon. So here's a few. Are all lab procedures BSL-1?
So you're talking about safety here. So we didn't look at really disposal or any things like that for our cultures yet. So let's talk about what students grow beyond the 3 microbes we provide them with and how they handle all these microbes. So first of all, we've already seen the 3 microbes that we provide students, which are non-virulent strains of those bacteria species and that eukaryotic cell. We do have a few labs where students actually plate environmental samples. Those environmental samples being from their immediate contact area, meaning their skin or their household surfaces. So we do not have students going out into the wild blue yonder to collect samples. When we do not have students collecting samples from, you know, the grocery store or even their workplace, for that matter. Any samples that students collect and plate out, they do not grow those microbes in a broth, by the way, unlike the ones we provide them with. Any microbes that students collect and plate out, they never open the plate once the colonies have developed. They are merely describing the morphologies of the colonies that are present. They are not manipulating those plates in any form or fashion. And then not only those plates, but also the cultures that we provide students with at the end of every exercise, except for the mother cultures that students maintain of those microbes we provide. But the microbes used during every exercise, students are soaking in bleach, undiluted, for a minimum of 2 hours before disposal. So we know students don't have autoclaves, obviously, like we use on campus, but the volume of microbes and the volume of spent media that students are disposing of is very, very small. And again, soaking in undiluted bleach for 2 hours will mitigate any risk from those organisms. What else did you have, Audra?
Plenty. I'm going to try and get through them as quickly as I can. I know we're on a time crunch here. Professor Basil asked, perhaps you can address this at some point. Can you add a requirement that students pass the lab safety section before starting labs? That's
so all of those prerequisites. So let's go back to the home page. So all of these prerequisites are set up exactly like the labs we're taking a look at, meaning they have 3 sections to each one. So exploration, the exercises, and an evaluation. The only way a student is even able to get to the next prerequisite is once they complete all sections, including the final exam for each of these labs. So I can't guarantee that your student passes it. That's really on them. But I can guarantee they have every opportunity and multiple times over to gain the knowledge that is desired, that they understand by the end of each of those prerequisite labs. Now, having said that, we'll look at some instructor tools here in a moment. And you have an option as an instructor to unlock labs as you determine your students are ready for. So you have the ultimate control over that, but we try to build in as many, anti-fail mechanisms as possible as far as ensuring that students understand the information in these prerequisite labs. That's a great question. Thanks, Audra.
One more before we move on, and then we can come back to the rest of these towards the end of the presentation. We have a huge outpouring from the community here, our attendees, to see the V-scope in action. Can we do a quick demo of that?
Yeah, we will. Let's quickly go through an instructor. I'm not going to spend as much time on the instructor side of things, unfortunately, so we will have time to preview the V-scope if that's okay with everyone. So, and I think this is a good hard stop, Audra, for the questions, and we'll resume the questions at the end of this next part of the presentation. So thank you for your questions so far, everyone. Keep them coming, and we'll get them answered, I promise. I just don't want to force everyone to run over our time allocation today. so I can get through the presentation, and I'll stay as late as you need after that. So let's look at Instructor View. And you'll notice the instructor landing page is a little bit different, and that there's a place to add comments. So you can add welcome comments for your students, and they'll see that on their landing page as soon as you input that and hit save. Here we see that you control as an instructor which labs are locked and unlocked. Now, you cannot control which labs are prerequisites or not. Those are set up by us. But beyond the prerequisite labs, you can change the order of any labs. You can hide labs from student view. And then again, you can control when students enter any given lab. So this allows you to do a lot of things. It allows you, to assure that the laboratory course flow of topics mimics your lecture flow of topics. So you have total control on the order of the labs in your student's lab manual. Again, you can select to have students sequentially complete lessons, and that's this box here. And what that means is a student can't, in this instance, begin antibiotic sensitivity until they've completed hand washing. Or you could take that off, and then if we went to student view, these would all be unlocked and they could move about however they wanted. And then if you click this, your students never see the hand washing in normal flora lab until you make it magically reappear by clicking the button. And if you're ever wondering what your work is doing on the student end, what we're in today is an instructor account. And so you as an instructor can toggle between student view and instructor view on the fly. So you can see in real time what your changes to a course are making on the student side of the experience. So let's take a look at a lab in Instructor View, and then we're going to show you specific tools beyond that you have, but I want to get through this as quickly as possible. The main difference in a lab in Instructor View is that, one, I mean, all the questions and stuff are still there. We've seen this, and I don't want to take up our time to do it again. But the main difference in Instructor View is that multiple times on every content page, you will be presented with this pulsing blue bar. And if I hover over it, it stops pulsing, and then I can click the Add button, it's a little plus symbol, in the right-hand margin. And when I do that as an instructor, I have the opportunity to add supplemental text, images, and videos live into my student's view. This really is a great tool for you to customize content to the needs of your course and specifically the students in your course. So you can upload just that, any kind of text, any kind of images, and any kind of video files as long as they're MP4 formatted, and those will appear and play for your students when they encounter them. And here's what that looks like. So let's go. And then as soon as I hit save as an instructor, you notice that it appears in a different color. And if we were to go into student view now, the student would see any content I've added as instructor highlighted on each one of their content pages. So their focus will automatically be drawn to anything that you've uploaded within one of the standard lessons. And if any point in time you wanted to delete that, you could go in, edit it, and delete it, and then it would disappear from your student's view. So, and you have the opportunity to do that not only on all of the background pages, so you'll see there's more pulsing blue bars, but you also have the opportunity as an instructor to perform this in the procedures. So if you wanted to add a supplemental video, now like this particular procedure already has a pouring egg or plates video that we've provided for your student. But if you wanted to, if maybe you had set some things up in the lab for your on-campus students and you wanted to film that and share with your online lab students, anywhere within these procedures, again, you can click the blue bar and upload a video of your choosing, or a static photograph, or again, more text boxes. So again, a wonderful tool for you to customize the student experience. The only thing different you as an instructor sees when you click the data tab is what we've uploaded for the answer key. So whereas when students click this, they just see response panels wanting them to put data in. As an instructor, when you click on that, you actually see an answer key. So that's the most basic instructor tools, and what's really different from a viewing standpoint within a lab. But we've given you far more tools than that. First and foremost, we've given you a dashboard for all the students in your class, so you can see their progress throughout the semester. So this class has some pretty famous people in it. You know, we've got Snoop Dogg and his pal Martha Stewart. We've got Betty White, who's saying that even though she's mid-90s, it's never too late to learn, and a few other famous people here. So we can see here that this class has progressed to the last lesson, and Snoop has actually got started on the last lesson. We provide you, an educator, with a legend, so you understand what the colors mean here. White means people haven't started it yet. Green means the work has been submitted, and that you can now go in and grade that. And then again, you can look for each lesson individually and see how many students have progressed as a bar graph and how much time they spent on each section of that lesson. So that's your course dashboard. We've seen the lessons tab. The materials tab, that's the same thing that a student views, so no different. Documents, this gives you the opportunity as an instructor to upload additional documents above and beyond the ones that we've already placed on there for your students, if you would like to put them here versus live within the content. We do offer you downloadable answer guides, but better than that is using the integrated Gradebook tool. And so here we see Gradebook. Here's our 5 students in the class. We've got Snoop and Martha and the other folks. And then here are all the lessons and the point weight per lesson. Now this is up for you to decide as an instructor of how much importance you want to give a particular week or lesson within the course, prerequisites versus regular lessons. It's totally up to you, and it's very customizable. All you do is select a lesson title, and then you're provided with a list in order of every question that a student encounters in that lab. And then you can go in and change the point value for each of those questions as you see fit. You know, if you don't want to grade data tables, you don't have to. If you don't want students to be graded on those formative drag and drop assessments at the beginning of every section, and I encourage you not to grade those, then you can go in and mark this as 0 per question. Anything you change on here from the standard 1 point per question that comes with the labs, those changes are saved, and every teaching term thereafter, they stay that way unless you go in and alter them again. So this is how you set up the auto-graded questions and how much they're worth, and it also how you set up the point values for the data tables and those open-ended end of exercise questions. So once you've done all that and you've got everything the way you want, you can actually then go in and assign grades to your students. So here we see Snoop has been all over the board. He did so-so on his V-scope, Pretest, a lot better on his staining lab. And then if I want to go in and manually grade his open-ended responses to his experiment questions, I can do that as an instructor. So I just click, I'm provided with the integrated answer key here, and then I can see the student's response. Actually, Snoop didn't answer that question. And then I can leave a comment for my student saying no answer provided, and I'm going to give him 0 credit for that. And then my comments to him will be visible in his view when he logs back onto the platform. But again, you've seen these answer key images already. They're integrated into the Gradebook grading tools. A very straightforward way to assess your students. A couple other things, and then we will look at the Vscope. So, what else can you do? What other tools are available to you on our platform? One tool is custom lesson authoring. And this gives you all of the authorship capabilities that I have and my team has when we build content originally on the Lab Manual platform. So it allows you to create supplemental lessons to the lessons represented in our kits. It allows you to build final overall comprehensive exams for the whole semester or practicals for after every few lessons. But basically, any type of content that you've seen that we've built on the platform, you can go in and create lessons with all those same content building tools. So, and then you can intersperse those lessons within the lessons that we've already pre-configured on the platform for you and your students. Lastly, settings, and then you saw the very last box is the Vscope, and I know that's what you're waiting for. But settings just shows you how to share access to the lab manual with your students. Now, the most basic way is simply copy and paste the student enrollment link into your learning management system or into your syllabus, however you communicate with your students, and then your students gain access to their lab manual. A much more sophisticated way of doing things is to integrate our lab manual content into your learning management system. And you can integrate that completely, meaning that you, on a lecture page within your learning management system, can embed the lab chapter that corresponds to that lecture page. So your students then aren't logging on to this platform per se and selecting a lab to complete. They are completing the lab as it occurs adjacent to your lecture within your learning management system. So truly a single sign-on experience for your students. It's a seamless laboratory experience for your students, meaning that they're not having to remember multiple sites, multiple passwords, or anything like that. All of those gradebook functions that we saw are also fully integratable into your learning management system. So whether you grade on our platform or you grade on our platform that's embedded in your learning management system, all those grades port automatically into your school's gradebook tool for all of your students. So it can be as seamless as you would like it to be. There's no charge on our end for any services in assisting you to integrate systems. We're happy to help in any way. So, without further ado, let's take a look at our Vscope program. So Vscope stands for Virtual Microscope. We did develop this platform in-house. It lives seamlessly inside the lab manual. If you as an instructor choose to have your students use a virtual microscope instead of a physical microscope, they will have access in the drop-down, like we just saw you have access, in the drop-down from the home screen of their lab manual platform. Now, this is not simply click on an image and see a picture. This is a functioning microscope, meaning students do have to perform a sequence of activities in order to produce a viewable image. And we give them a cheat sheet in the lower right-hand corner, meaning they have to move the stage to its lowest position and turn on the illuminator and select the lowest power objective lens before placing a slide on the stage. So we will do just that. This is ADA compliant, meaning I can use a mouse like I'm doing now, or I can use the up and down arrows on my keyboard. If I want to know how to do that, I just click this legend here. It gives me a pop-up and shows me how. So I've lowered my stage. I'm going to turn on my light source, my illuminator. I've already got the turret dialed to the 4 power objective. And now I can go find a slide. So this is my virtual slide library. This is agnostic for all of our lessons and all of our disciplines. Having said that, our titles are very specific for each slide, and it is exactly how the title appears in our curriculum. So as long as a student is following the lab manual instructions, they will know which slide and easily identify it in alphabetical order here in the slide library. So here we go. Let's do a whole amount of the yeast cell. So you notice when I first click on that and it's on the stage, on my viewing window, which is in the middle right-hand half of my screen, it's all blurred out. Not unlike if I had put in a slide on a microscope stage originally. So what I need to do is I need to center my image and I need to raise my stage until it comes into focus. So I've got stage adjustment knobs here. Now we let the student cheat, if you will, and then we give them a green check mark when they've accomplished the ideal centering, even though it's still not truly centered. But nonetheless, we want to at least give them some kind of confirmation that they've accomplished this particular step. Now, there are numerous slides where the ideal cell they need to locate is not in the center of the slide, and the students will be panning around. But for the sake of this demo today, we're just going to follow the instructions as they are on the checklist here. So we're going to raise the stage up now to start to bring things into focus. And I probably overshot. We're getting close. We can always use the fine focus knob then. Oh, that got closer, and I'm going too quickly. I find using the arrows on my keyboard does this a little more precisely. So that's looking pretty close for low power. And now I can go in and I can change my objectives. And there's high power, and then I can do a little fine tuning if I need to. And if for some reason that wasn't centered, I still have the opportunity to do that, or I can pan around and search for a particular group of cells. When I'm satisfied as the viewer with my end result, I can click download image, because you may recall when we were looking at that morphology and staining lab in the student view, we were being asked to upload our final slide images as proof that we had accomplished the main goals for that lab. And so if I'm a student using the V-scope, obviously I can't look at the slide I just made. I've got to look at a sample slide, which this is one of, and I have to click download image. And when I click download image, you may see at the bottom of my screen now, I get a JPEG, and now with that JPEG, I can drag and drop into the response panel in my lab manual itself. So very, very easy for me to do as a student. Again, I have access to all slides in the library. As a student, I just need to pick the right one based on the instructions in my lab manual. If I'm a student using a real microscope, any prepared slides that come in my kit, they're going to be the same title for those prepared slides as are called out in the instructions. And in fact, we developed our slide library for this V-scope off of the very prepared slides that we send students using real microscopes. So there's no difference whatsoever. So if you were to teach one semester using the V-scope and another semester having your students use real microscopes, in your answer key, you would see exactly the same images. So again, our content is microscope agnostic, not simply from the standpoint of physical microscopes and the ones we provide versus ones students may already have, but also between the V-scope images and real microscope images. So hopefully that's a good preview for you. I will let you know, and I'm sure the panelists today who are answering your questions that you're chatting will let you know that if you are interested in our curriculum, including our vScope, we do grant demo access to the curriculum, and you will have plenty of time to investigate exactly how the vScope works, the slides that we have in our library, and our curriculum itself. And then keep in mind, if we go back to, oh, I didn't want to do that. If we go back here to the live instance, somewhere, if we can get there, taskbar, there we go, that using the vScope is a prerequisite. So if you've chosen for your students to use the vScope instead of a real microscope, they will have a training tutorial before they ever need to use the vScope of exactly how it works. They'll get to practice focusing on the images that they select in there, downloading the JPEGs of their focused images, and then uploading those JPEGs into the image panels in their lab manual. So again, that's a prerequisite. Like any prerequisite, we want to set your students up for success before they ever begin their first real lab. So using the vScope is no different than the other prerequisites. So lastly, I talked a little bit about ADA compliance regarding the vScope, but all of our curriculum is, meaning those drag and drop questions, those interactive multiple choice questions, the content itself, all that stuff is screen reader compatible. Any embedded images have supplemental text documentation. All embedded videos are closed captioned. So I know your school has to meet ADA requirements, including for its online courses, and I want you to be assured that the curriculum that we build in our platform are both meeting of those guidelines. So that concludes the demonstration today. As promised, I want to get done in our 1 hour allocation. I know you have more questions, and I'm happy to stay online as long as needed. Certainly for anything that our panelists couldn't have answered for you in chat. But again, thank you very much. I know the thought of teaching micro online is intimidating, but we want you to rest assured that we've been doing this for a really long time. One thing I didn't talk about, I know you had some safety questions, but we do provide, and I'm sure the panelists have answered this, we do provide liability coverage for your students using our kits in their prescribed fashion. I mean, we are confident that the materials that we provide and the instruction that we provide for using those materials is the safest possible for conducting microbiology experiments off campus. And to that effect, should your students have a mishap or otherwise, again, we are the liable party, not you or your institution. So thank you for your time, and then I'll stick around and answer any questions, and I'll turn the closing comments over to Audra.
Bravo, Dr. D. This is so fascinating. I want to say that this webinar probably has been the most engaging webinar we've had ever in the history of the company. Just in the Q&A function alone, we had 100 questions come through. So the reps were able to answer the majority of these, but I want to go back and circle around to the ones that they left for you to answer. A lot of concern out there from the instructor community about the cultures themselves, how to dispose of these cultures. Some universities are saying, hey, we won't even allow students to work with cultures at all. So if they're included, game over. Can you speak a little more about the cultures that are provided and the safety precautions that we have put in place in order to keep the students safe?
Well, again, we've covered that there are non-virulent forms of Staph aureus, E. coli, and Saccharomyces cerevisiae. You know, if your university doesn't allow students to use cultures, i.e., they're requiring virtual-only microbiology, then very sincerely, we're not a good match for you. You know, and if that's determined your university side, then I really don't have a solution for teaching hands-on microbiology without microbes. So, I'll plead ignorant on that one.
Okay.
But again, as far as disposing of cultures, soaking in bleach for 2 hours, the amount of material students are using for any procedure is very, very small. You might have seen even that Congo red stain. Students only get 1 milliliter stain. And yeah, they get more of the gram staining, multiple milliliters of each of those. But again, those can be used throughout the semester. So at any given time, they're disposing at most of 3 or 4 slides that have been heat fixed. They're disposing of a plated culture that's been taped shut and soaked in bleach for 2 hours, or they have a spent broth vial. The volume of broth in vials that we provide students with is 5 milliliters. So very, very small amounts of culture. And again, soaking in bleach neutralizes those within 2 hours. Again, I understand your fears, and I guess the only thing I could say to you, if your administration has concerns, is again, we are the liable party. We assume all risk. So, and I'm sure our representatives in the proper departments can forward any kind of those statements to you to share with your administration if needed.
Thanks, Dr. D. There was a question that's sort of applicable to what you're just talking about from Professor Christine. If they are using the vScope, what's the incentive for the student to make and stain a slide?
Well, the incentive is they're required to upload an image with their name and date stamp on it that they actually made that slide. So if they're not uploading that image, and then it's pretty indicative they didn't, and you as an instructor know they didn't do it. So, so again, a vScope is a solution we offer for you and your students. Again, most schools choose it from a cost-saving standpoint because so students don't need to purchase a microscope. Would I teach on campus with a virtual microscope? Absolutely not. I'll be the first to say that. This is strictly an artifact of online education. But again, if you don't want students to incur the cost of attaining a microscope that's at least 1,000 power magnification in conjunction with an oil immersion lens, then we offer the vScope as a solution for you. But we make sure that anytime a student has to create a slide, that they take a photograph of their creation. So at least you have proof as an instructor that the student actually did make the slide.
Another question out there was, someone was hoping to hear the answer about which kit to buy if the course is offered for 3 days per week for 6 weeks in the summer. I know that your rep could help you with that. But the second half of that question is, with this current pandemic, it might be hard to get bleach. Is there any alternatives to bleach?
Well, I don't know that any sanitizing material is necessarily more readily available right now than not. But yeah, I mean, even a stronger alcohol, stronger isopropyl alcohol would be fine. But again, that's kind of a difficult thing to answer. Certainly, any germicidal compound would work. Bleach has always been the most cost-effective and readily available for students. And yes, these are challenges and times we live in. If a student can always reach out to student services if they find themselves taking the course and having a kit and needing disposed microbes, and they suddenly can't find bleach or alcohol, and we will guide them accordingly at that time. But I don't have a crystal ball right now to tell you that you can go out and buy glutaraldehyde. Not that you could buy that anyway if you weren't on the technical side of things, and be guaranteed to kill your microbes. So we're happy to take that on a per case basis with any given student. But again, yes, these are challenging times, and because of that, I don't have a fix-all as far as one compound I can tell you will always be available.
Thank you. Several questions out there about incubation of these cultures. How are they incubated? Someone even asked, how do we keep the temperature at 37 degrees Celsius for incubation? So can you speak a little more to that?
Sure. So all the three species of microbes that we provide your students can be successfully grown in sufficient numbers within 48 hours, as long as the temperature is over, 22 degrees Celsius. I know that's not ideal, but it does work. And so when I look at what we've done over the decades as a business, in our early days, we had students trying to construct homemade incubators, which in hindsight could easily have been a fire hazard, and the results were not consistent with what the students were doing anyway. We've worked with our vendor who provides us with the strains of microbes that we use to locate strains that will grow readily, at lower temperatures. In my experience, the E. coli strain that we're using is a vigorous grower, and even at 22 degrees C, I have really noticeable turbidity in a liquid culture after 24 hours, and I do have visible colonies within 24 hours. We tell the students, and you saw in that time allocation in the staining lab, that if you don't have a developed culture set aside 48 hours, it's kind of a fail-safe. But again, with the microbes we provide, 24 hours should give the student enough microbes that they need for any given procedure, whether it's, as you see the lower title on this very succinct list of labs, an MRVP test. Again, they should have enough development in an MRVP broth within 24 hours of inoculation. So anyway, but no, that's a great question. And again, I know being an on-campus instructor, yes, we're using lab-grade incubators, and we can develop E. coli cultures in within 18 hours easily. But rest assured that worst case scenario, as long as I can keep my temperature at 22 C or higher, within 48 hours, I have all the microbes I need for any procedure with those cultures that we are providing.
Thanks, Dr. D. So many questions about the V-scope. There's one about unknown microorganisms. So typically at the end of the semester, students are given an unknown culture to identify using all the protocols and biochemical tests to identify their unknown microorganism. Is there anything similar to this in our experiment library that we offer?
Yes, we do have a testing for unknowns. So it's not in this little list here, but what students are doing is they're using differential media, and they are using, they're growing both the E. coli and the Staph epi in the selective and differential media types. And at the same time, they're growing two samples collected from their bodies. So not even from the environment, but from their physical bodies. And they are comparing what these two unknown samples are doing on these selective and differential media to the two knowns. And they're trying to draw some conclusions of what they could be based on the results of those tests and again, the comparison to the knowns. So not quite what you do on campus, meaning we're not sending a student an unknown lyophilized tablet, but they are going through the protocols that you would use on campus to identify an unknown.
Cool. The V-scope, can instructors add additional slides that aren't in the library already?
Not really. So unfortunately not. So those images in the vScope are very painstakingly taken, meaning it's not simply me at my desk using my video microscope camera and doing one shot through of what I'm seeing at any given time. I actually have to take dozens of images at all magnifications and then forward those to our software developers because that's how students are able to pan around on each of those slides and isolate different cells or cell groupings and stuff on a slide, which they would not be able to do if I just took one static image through the camera lens. So for that reason, I can't, the functionality that students have using the vScope, they wouldn't have if people just started submitting to me lone images. Is it possible? Yeah, it's possible, but it's a lot of work. And it's not something you could do on your own as just having access to the scope. So yeah, that's how I can answer that.
Great. A lot of these questions coming through are about the experiments in the experiment library, like what are all the stain techniques included in your lab manual? I have a question, concerning acid-fast stain and unknowns from Corey Burns. Is there a virtual oil immersion objective on the vScope? Can you answer any of those now, or should we just direct our friends here to their dedicated distance learning?
Yeah, so I would really encourage the panelists and their peers to share our either links to our microbiology lesson library or our webpage on that topic and things like that. And if there are residual questions after that, then reps can forward those to the science team at our company, and we're happy to address them.
I hope that I got all of these questions here. I know there'll be a few lingering questions out there that your distance learning specialist will be able to address. Dr. D, thank you so much for your time and expertise and knowledge and sharing your, just what you know and wisdom on our platform and our curriculum. What a wonderful webinar. For the instructors who are still here, thank you for your time joining in the conversation and engaging with our lab solution. We look forward to helping you in the future, and we wish you all the best. Stay safe, stay healthy. We will talk to you soon.